Thank you for that nice introduction. Okay, let me dive directly down into the subject area. I will start with a background for our general approach. So we are neuroscientists. We do intracellular recordings from neurons in various parts of the central nervous system. And for haptics, I think this is particularly important because actually haptics is not purely a cortical phenomenon. It also depends quite a lot on structures in the brainstem, in the spinal cord, and to some extent cerebellum and of course the thalamus. And we are, as far as I know, we are the only lab in the world that can do these intracellular high-quality recordings from all of these structures. So an example of such recordings are shown to the right. You see a patch clamp pipette that is attached to a neuron, and then you can record intracellular signals, and you can provide the different kinds of input, and you will get different kinds of responses. Now, we study the sensory motor systems, so with a focus on upper limb. So it is not just haptics, it's also a little bit motor control, but of course the subject area of this presentation will be the haptics. Now, these recordings, they require that we do animal recordings, first of all. This cannot be done in humans. And also they required, in order for us to be able to make this recording over a sufficient amount of time, they require that the animal is somehow immovable. And we do that through decerebration when we study structures below the cortex, and using light anesthesia when doing the recordings inside the cortex. There is sometimes rising the point, why do you not do these recordings in the awake animal? My general answer to that is that in neuroscience, we are very far from the level of knowledge that we could actually make use of and correctly interpreting all the complex information that's flowing in an awake animal. There still is a lot of infrastructural information that remains to be discovered when it comes to the organization of processing of haptic information in the central nervous system. So that's why we are having these approaches. Now, this figure, it points out two specific structures. There is the cuneate nucleus, which is located in the lower part of the brainstem. So it's a 3D image down here. You will see the green structure, that's the cuneate nucleus. So we will start out in that location. And then we will move on to the neocortex, which of course is up here. Okay, first of all, I would like to start out with pointing out that haptics involve large populations of tactile sensors. This is a histological image of the skin. This is a cartoon of the same skin with some skin sensors located within it. And of course, every time we do some kind of interaction with the outside world, even with very pointed objects, there will inevitably be a spread across large parts of the skin of these mechanical effects that the tactile sensors are there to detect. So basically, to make a long story short, whenever we make contact, there are the laws of contact mechanics that apply, and they stipulate that there are non-local effects inevitably. So not just the actual area of contact between the skin and the object is engaged, but also large areas around it. And with this kind of conceptualization, it differs a lot from the pixel-wise scheme that I would say is otherwise dominating the neuroscience literature on haptics processing. So with pixel-wise scheme, I mean, of course, that there's a lot of focus on the properties of individual sensors. We instead take the perspective that inevitably there's a large number of sensors being involved in every single, even the simplest form of haptic interaction. Now, this results in that all these skin object interactions, they can be decomposed into haptic input features. And what are those? Well, in order to study that, we, together with Vincent Hayward, who manufactured this excellent device for haptic stimulation, we made recordings within the QNH nucleus. Starting out with the device, it gives you the ability to emulate some of these haptic input features or haptic input dimensions in isolation. So to not spend too long time on this slide, I will just jump in here. You will see now we recorded from two different neurons in the QNH nucleus, neuron 1 and neuron 2. We provided through this haptic device or haptic display different kinds of inputs. So some of them emulate the effect that arises when you make an active contact with an object. You will see that this effect plays out over a relatively long time. And again, it doesn't engage a huge number of tactile sensors distributed in different parts of the skin. You can see that these two neurons, they responded both to the contact on, but in very different patterns. So we will be coming back to this let's say, response pattern issue very much in this presentation. With another kind of haptic input dimension, now there was only one of the neurons that responded, and whereas for another type of haptic input dimension, there was the roles between the two neurons were flipped. So now only the green neuron responded. Now we did this kind of analysis for a relatively large number of neurons, and we had 18 inputs, so basically 18 haptic input features in principle or potentially, and we could combine the activation across this population of neuron for each individual repetition. And you will see that the symbol, both the size and the color code, indicate which of the input was delivered. And already after 15 milliseconds, there was actually only using these 15 neurons in the QNH nucleus, there was a pretty good picture of what was going on. And then after 150 milliseconds after of these ongoing stimulations, the population of neurons could tell every single one of these 18 inputs apart. Now, this is the combined population effect across again, a very small population of neurons in the QNH, 15 neurons in this case. In a human, there are tens of millions of these neurons, so the capacity to separate the inputs are pretty high. If we look at each individual neuron in the QNH nucleus, and we project their position depending on their response selectivity to each of these different 18 inputs, and then we made a multidimensional scaling, we would see that each neuron, which is now a sphere in this plot, they have a very different position. So if we can provide rich spatial temporal patterns of sensory activation, which was enabled with this haptic display device that was developed by Vincent and colleagues, then all of a sudden, this population of QNH neurons, they turned out to be highly specialized representations within each individual neuron. So this is as opposed to traditional neuroscience approaches to tactile stimulation, where researchers want to have as reduced input as possible. I'm saying that when you use those reduced inputs, you risk missing the main point, which is that the brain really wants these complex spatial temporal patterns of skin sensor activation. Only then can you sort of get to grip with the actual organization of the neural processing of haptic information. All right, so this is still in the brainstem. When we look at the formation of percepts, we inevitably have to go to the neocortex. Now, this figure shows you 4 different kinds of illusions, perceptual illusions. I'm sure you have seen at least a few of them before. So the top left, you see there is an image of a cat head, and if you focus on that, you will get the impression that there is something in this image that moves. In reality, there is nothing that moves in this image. There are just the right set of cues to trick the neocortex to believe that there is movement in the image. The top right corner, I think, is maybe more familiar, and there you would see that the square A and the square B, they actually have the exact same gray shade. I think even when I tell you, it is impossible for you to convince yourself that this is the case, and this is because of the perceptual mechanisms that reside in the neocortex, which takes a large set of surrounding cues in order to deduce, in this case, the shading of the gray. The lower left, you will see another visual illusion. The bar has the same color, but you will see, depending on in which field you look, you will perceive it as having green respective red color. And in the lower right, we see a haptic illusion, which is the comb teeth illusion, where you drag a pen along the teeth of the comb, and then you will get an impression that your finger sinks down into the comb. All right. Now we're going to try to deduce some of the mechanisms that underlie these effects. And then we have to go to the neocortex. But of course, when we go to neocortex, we should not forget that the haptical functions of the brain, they very much depend on control of the muscles. So there is always activity in the neocortex, and that will always impact the activity in the spinal cord. The spinal cord in turn controls the muscles, and there are a huge number of sensors even within the muscles. And whenever we have some kind of contact between the skin and an object, we will then engage also large populations of this, the skin tactile sensors. Now, all of this sensory information will be provided back to the spinal cord and actually contribute to updating the motor command. But of course, here it will be mostly interesting because all of this information is going up to the central nervous system, and the brain has to somehow make sense of all of this. And we are going to have a look at the processes that underlie that processing. So, first of all, the perception must be defined by internal expectations. We can see that through a variety of, let's say, basic observations. So, the same object, when we interact with it, will never generate the exact same spatial temporal pattern of skin tactile or skin sensor activation. So, there are a lot of differences that arise. Given that we have such huge populations of sensors, even a small change in exact skin location will give you a relatively different spatial temporal pattern of skin sensor activation. And of course, the type of movement or just the amount of tension that we apply between the skin and the object, it will also vary, and it will vary over time. But it will most of all vary between different instances of skin-object interactions. There are also a number of noise factors. So, but even though each skin-object interaction is unique now in terms of the skin sensor activation pattern that the brain receives, still, despite this variation, the perception of the object is constant or invariant. So, and there is also a number of phenomena that indicate that there is a lot of object completion going on. So there's a circuitry coding of which type of skin-object interaction is expected to take place. And this object completion procedure can have different names in the literature. So if you're interested in Bayesian inference, you call it priors. You can call it expectations, or you can call it predictions. But it's all a sort of one and the same thing. And that's what we're interested in finding out. So to do that, we teamed up with the Biorobotics Institute in Scuola Superiore Sant'Anna in Pisa, and they have a robot fingertip by which we could emulate different kinds of skin-object interactions, and we could now obtain a set of spatiotemporal skin sensor activation that we applied to the distal skin of digit 2 of the rat. Now, here is the point that we have 8 different kind of input patterns emulating different kinds of interactions, but we can repeat each single one of them perfectly for a very high number of times. And it will be the exact same spatial temporal pattern of the skin sensor activation. So now when we are recording within the neocortex, any variation in responses has got to be due to internal factors within the central nervous system. And here is an example recording. We first recording S1 cortex in the rat in this case. You will see this is the same neuron. So we can, in this case, it's a layer 3 pyramid. When we present it with one stimulation pattern and we repeat it a high number of times, you will see that the response is never ever exactly the same. Now, even though the skin sensor activation pattern is exactly the same. So there's a lot of internal variation also here. When we provide another pattern, you will see you have the same variation, but overall the differences between these two, the responses evoked by these two different input patterns, one can sort of intuitively feel that there are such differences, systematic differences. And indeed, when we do principal component analysis of these responses, and then have a k-nearest neighbor classifier for the responses when they are sort of located in that principal component space, we see that the signal, the intracellular signal, with a high degree of accuracy, reports which of the stimulus that was actually being provided. This is across 100 repetitions for each stimulation pattern, so in total 800 stimulations, since we had 8 inputs. Now, this confusion matrix you will see here, are going to use this a lot in the next few slides. So first of all, when you look at different neurons within the somatosensory cortex, and we provide the exact same input, the tactile input patterns, we will see that each neuron generates a unique response pattern, or at least to some extent unique response pattern. This is highly interesting because it means that each individual neuron that we recorded from, they did take this input, but they did encode it in a different fashion. Now, this could be explained by that each neuron has a unique position within the global neocortical network, and therefore they will pick on different aspects of the information that is being provided from the skin. Now, this complementary decoding by a population neuron is potentially highly useful for high precision decoding, because if we combine the contribution of just a few individual neurons, we will get very high precision decoding indeed through the cooperative decoding between these highly responsive S1 neurons. Okay, so the idea of complementary decoding is another main idea that we will have in the next few slides. So, but before we move on, one very surprising finding that we had in this early study was that when we look at the different assumed layers of the neocortex, so I'm saying assumed because it's been shown indisputably by anatomical methods, but there was never any good functional evidence that these are sort of functionally separate entities. And what we find is that when we look at neurons located in these different layers, we do not really see any difference between them. In this case, we explored that for many different parameters. One was the average decoding accuracy. In this figure, we show the maximum difference between the input that was best decoded and the one that was worst decoded. So we saw that some neurons, they really preferred some patterns and not others, and then you have a high value here. But even sort of with this discrepancy decoding, there was no, we could not find any really good evidence of layer specialization. It's important, but here is another important aspect. So even we then move across different parts of the neocortex. So this is a neuron recording the primary visual cortex, provided with these tactile input patterns. And you would see that this is the neuronal response, one neuronal response to the S infinity pattern. You have the spikes in the raster plot here. You have a peristimulus histogram, and overlaid you have this kernel density estimation of that response. So for each input pattern, we get is a kernel density estimation of the response, and here at the bottom we overlay them. And you can see that these neurons, or this one neuron, it has a very good capability to tell these tactile input patterns apart, even though it is located in a primary visual cortex. Many neurons in the primary visual cortex do not have very strong responses to the tactile inputs, but they do have some responses. And when we moved on and we compared a number of different neurons in the visual cortex, we could see that this principle of complementary decoding, now across these five neurons in the primary visual cortex, it was there also in the primary visual cortex. So each individual neuron within the visual cortex and any other region of the cortex that we explored, they do have some kind of unique contribution to the population level coding of the tactile input patterns. That may look surprising at that point of time when we studied this, but actually this is an old tenet. If you move from one neuron and you move 3 or 4 synapses away, you will end up just anywhere within the central nervous system. And moreover, there were more recently, let's say, a more exhaustive anatomical analysis of these neurons, and it would turn out that we have for a very long time been underestimating the full extent of the distribution of the axonal tree. So this is a neuron in the primary visual cortex that projects all the way forward to the prefrontal cortex and also to the motor and sensory areas. So there's sort of infrastructure for these observations that we just made. Now I could see motion turning up, so I guess this means that I have very little time left. I would then try to take a minute.
Sure.
Yeah. So because now we're coming to maybe the more interesting aspect. So when we provide one of these tactile afferent input patterns and we make these recordings, we can see that there are a multitude of specific solutions provided in this neuron for each given input. Now, this is interesting because this means that of all the synaptic inputs that this one neuron receives, they will enter different responsive states depending on probably the sort of the internal expectations that is resident in the network. I will not spend much time on this. I will just also show that when we have a cortical stimulation very far away from the somatosensory cortex and we induce a subthreshold stimulation, we can obtain controllable perturbations of the network solutions that is provided by the network. And that is shown in this relatively complicated or large confusion matrix, where you see here we have the tactile input patterns that I just showed you, and we can see as before that the different network solutions, they generate responses that to a relatively large degree is unique. When we combine these tactile afferent input patterns, the exact same ones, but now with the cortical stimulation, we all of a sudden get a complete different set of responses or network solutions within the cortex. Okay, so I'm running out of time. I will then not